The overall goal of this isolation procedure is to enable live cell imaging of the proliferating cells that are undergoing interkinetic nuclear migration in the retinal explant cultures from damaged zebrafish.
This method can answer key questions in retinal regeneration, such as what mechanisms drive interkinetic nuclear migration, or whether Mueller glia and neuronal progenitor cell interkinetic nuclear migration are similarly regulated. The main advantage of this technique is that the dynamic behavior of the proliferating cells undergoing interkinetic nuclear migration during regeneration can be monitored, which permits the investigation of cell cycle stage-specific mechanisms.
So this method can provide insight into interkinetic nuclear migration. It can also be applied to other retinal research areas, such as investigating microglial behavior or Mueller glia phagocytosis of dying neurons.
Begin this procedure by transferring the zebrafish onto a dry paper towel. Then, remove one of the eyes with a pair of curved forceps and transfer it onto a fluoro dish. Under a stereo microscope, orient the eye with the pupil facing the cover slip of the fluoro dish so that the optic nerve on the back of the eye is visible. Then, remove the optic nerve as close to the back of the eye as possible. In addition, remove the connective tissue outside of the eye. Hold the eye between its nasal and temporal sides with a pair of forceps while making an incision.